Hi, my name is Morgan Robinson and this is my exam video for Exam 1. In this video, I'll be defining the Lion hypothesis and describing the relationship between X inactivation and X-linked inheritance, including the role of bar bodies. X inactivation is when inactivation occurs randomly for one of the two X chromosomes in female cells during some point of development. The inactivated X chromosome is what is known as a bar body and can be found at the rim of the nucleus in female somatic cells. X-linked inheritance means that the gene causing a certain trait is located on the X chromosome, and the trait can either be dominant or recessive. An example in nature where we can see this hypothesis occurring is in tortoiseshell cats. X inactivation only occurs in cells with multiple X chromosomes, which explains why only female cats can be tortoiseshell. The coat color of cats is an X-linked trait with a big B producing a black coat and a little b producing a yellow coat. Therefore, a black cat would have a male genotype of X big B Y and a female genotype of X big B X big B, while a yellow cat would have a male genotype of X little b Y and a female genotype of X little b X little b. The genotype for tortoiseshell is X big B X little b. If we crossed a black male with a yellow female, we would see all the males having one X with a little b and one Y, making them all yellow. For females, one X would have a big B and the other X would have a little b, creating the genotype for a tortoiseshell cat. Now we will consider a second trait in cats, which will be hair length. Long hair represented by a big H is dominant to short hair, represented by a little h. For this example, we will cross a yellow short-haired female with a black male that is heterozygous for long hair. The female's genotype will be little h, X little b, X little b, and the male's genotype will be big H, X big B, Y. To start off, we will need to draw two Punnett squares to better understand the crossing of these traits. The first will be for color and will look like this. The second will be for hair length and should look like this. With this, we can draw a branch diagram that will show us the phenotype ratios we can expect to see in the progeny. First, we will start with sex. As we can see, half of the offspring will be female, while half will be male. Next, we will look at color. From the Punnett squares, we can tell that all of the males will be orange, and all of the females will be tortoiseshell. Moving on to hair length, half of our males will have short hair, and half will have long, and the same will go for females. Finally, we will combine all of our phenotypes. We will see that 1/4 will be male with a yellow coat and long hair. 1/4 will be male with a yellow coat and short hair. 1/4 will be male with a tortoiseshell coat and long hair, and 1/4 will be female with a tortoiseshell coat and short hair. If you've ever been around multiple tortoiseshell cats, you might be wondering why no two look the same. Well, even if a tortoiseshell cat was cloned, it would still not look the same as its mother. This is because the inactivation of the X chromosome occurs randomly throughout development, so the colors are randomly activated in some cells, while they are randomly deactivated in others. Therefore, all tortoiseshell cats look different from one another.